Thank you to the organisers for allowing us to present this work at the meeting. This paper is entitled High Hepatitis B Surface Antibody Titers Consistent Across 3 Lots of the Triantigenic Hepatitis B Vaccine Sci-B-Vac. My name's Adam Finn and I'm a paediatrician from Bristol in the UK. This slide contains disclosure information. Both my employer received funding to conduct the study at our study centre, and also compensation for consultancy work that I've done for the sponsor related to this study and this vaccine programme development. This is a presentation concerning an investigational vaccine that has not yet received regulatory approval either in North America or in Europe. This audience will be familiar with the problem of hepatitis B. This is the leading cause of chronic hepatitis, liver cirrhosis and hepatocellular carcinoma worldwide. And very large numbers of people, estimates ranging from 240 to 350 million people affected, of whom 5 million are in the European Union and European Economic Area. And it is estimated that as many as 2 billion people have ever been infected with hepatitis B. The initial infection is often asymptomatic, and therefore many people are unaware of the fact they're infected. In fact, the World Health Organization estimates that only 13% of hepatitis B infections in Europe have been diagnosed. And the incidence rates of hepatitis B in the countries that consistently report it in the European Union vary between around 7 and 10 per 100,000 in recent years. In the absence of any therapeutic modalities to treat this infection, active immunization is the main tool that we have available to us to try and control this massive public health problem. But the vaccines that we have, while effective, are far from optimal, and so there's a need to improve their immunogenicity, reliability, and protection. This study is a pivotal phase 3 randomized controlled blinded multi-center study that aims to compare the immunogenicity of 3 manufacturing lots and therefore demonstrate the manufacturing consistency for the triantigenic hepatitis B vaccine Sci-B-Vac, which is abbreviated to TAV, triantigenic vaccine in these slides. And we also compared the vaccine to a licensed monoantigenic Hep B vaccine, Engerix B, abbreviated to MAV, monovalent antigenic vaccine in these slides. A total of 2,838 subjects aged between 18 and 45 years in stable good health were enrolled 1-to-1 to 1-to-1 to the 3 different lots and to the comparator vaccine, and immunized at day 0, day 28, and 6 months, day 168. The primary endpoint of the study was consistency of the immune response across the 3 manufacturing lots in terms of the geometric mean concentration of antibody achieved 4 weeks after the 3rd vaccination dose at day 196. Secondary endpoints were to achieve non-inferiority in terms of seroprotection rates, that's greater than 10 milli-international units per mil at the same time point 4 weeks after the 3rd dose as compared to the monovalent licensed vaccine under study, and finally to monitor the safety and tolerability of the vaccine using a standardized approach to solicited adverse events as well as serious adverse events of medical significance as they may crop up during the 48 weeks after the initiation of follow-up. This slide compares the 2 vaccines. As you can see, there is a difference between the antigens contained, as mentioned already. The trial vaccine contains 3 different variants of the surface antigen, the standard surface antigen, as well as the pre-S2 and pre-S1 configurations of the protein, whereas the comparator vaccines just contains the surface antigen. In fact, the overall dose of the recombinant protein surface antigens in the trial vaccine is 10 micrograms, that's half of what is present in the monovalent vaccine. Both vaccines have an alum adjuvant, and the cell lines used to produce the vaccines are different. The licensed vaccine being produced in a yeast, whereas the Sci-B-Vac in a mammalian cell line. The pre-S1 antigen induces neutralizing antibodies that block receptor binding and therefore may enhance the ability of immune responses to this vaccine to protect. And both the pre-S1 and pre-S2 antigens can further boost T-cell responses to the S antigens, which may enhance the protection that the vaccine provides. This slide demonstrates how the study fell out. A total of 4,452 subjects were screened, of whom 36% were screened out, leaving 2,838 across 37 different clinical sites. And the table shows clearly that the randomization fell out very evenly in terms of age, gender distribution, body mass index, geographical distribution, and withdrawal rates and completion rates across the study. So we were fortunate in getting a clear and well-balanced randomization. So these are the first data presented in the talk. Please focus first of all in the red box, the three triplets of bars on the right-hand side of this, which demonstrate the geometric mean concentrations after the 2 doses and 3 doses of the vaccine, and show remarkable consistency, so achieving the primary objective of showing that manufacturing was consistently done. If you turn your eyes to the left of the figure, you'll see the equivalent GMC values for the monovalent vaccine, the licensed vaccine, which as it fell out were substantially lower, as you can see, both after 2 doses and 3 doses. Noting that the y-axis on this graph is a logarithmic axis of the mean titers in milli-international units per mil. With regard to the first secondary endpoint, predefined endpoint, which was the seroprotection rates and non-inferiority to the comparator vaccines, again, the trivalent vaccine under study is shown on the right, and the comparator vaccine in the orange bars on the left. And you can see that they are indeed not inferior, but in fact somewhat higher, 94% as compared to 51.5 after 2 doses, and 99.3 as compared to 94.8%, greater than 10 milli-international units per mil. So definitely achieved that endpoint. With regard to the second predefined secondary endpoint of safety and tolerance, this table shows the values of percentage of subjects that experienced various solicited as well as adverse events across the study. There are very similar rates for both vaccines, except for local reactogenicity, and you can see there are somewhat higher rates for the trivalent vaccine for injection site pain, site tenderness, and myalgia. And this is in accordance with previous experience with this vaccine. The one death in the study, which was in the study group, was in a patient with pre-existing hypertrophic heart disease. The death was not felt to be likely to be related to vaccination. So summarizing these data, the 3-dose vaccination completion rates were similar in the 2 studies, with no new or unexpected safety signals, somewhat increased reactogenicity as compared to the licensed vaccine for the trivalent vaccine, no new serious events of any sort that were thought to be vaccine related. And in conclusion, the lot-to-lot manufacturing consistency was confirmed. The non-inferiority of this vaccine to the monovalent vaccine was confirmed, and there were no new or unexpected safety signals. Looking back at the data in a post-hoc way, it's clear that the immunogenicity of the trivalent vaccine was significantly higher, 7.5 times higher after the second vaccination and 3 times higher after the third. And indeed, more than 90% of individuals vaccinated with the trivalent vaccine achieved seroprotection after the second dose, as compared to 51.6 with the licensed vaccine. So just to conclude, acknowledgement of our investigators, and many thanks.